Hello everyone, how you all doing? Today we will be discussing the metabolism of branched chain amino acid and their disorder. This is class number 44 and the concepts that we will be covering are how the branched chain amino acids look like, what is the chemistry, what is their common metabolic fate, how they are catabolized in the body and whether there are any disorders of this branched chain amino acid metabolism that are abbreviated as BCAAs. We will be discussing it all right. So, first of all, branched chain amino acid, whenever we are referring BCAAs, there are three: valine, isoleucine and leucine. Mind it is different from lysine, that is not this. Leucine, valine, leucine and isoleucine, they are all essential amino acid, means body cannot synthesize. Whenever we are dealing with an essential amino acid metabolism, we do not need to know how they are synthesized in body, because we need to consume them from diet. Whenever the rule is we are discussing a non-essential, we also need to know what are their sources. Okay. So, the metabolic fate of all these amino acids, right at the beginning let me discuss for MCQ purpose. Valine is a glucogenic amino acid, leucine is a major ketogenic amino acid, most important ketogenic amino acid that leads to formation of ketone bodies. Isoleucine has feet on both the boats, right? It is both ketogenic and glucogenic amino acid, right? And these amino acids actually, since they can produce either glucose or ketone body, they serve as a very important alternate source of fuel whenever there is condition that leads to deficiency of glucogen glycogen, that is starvation, right. So, there then the metabolism of these amino acids become very vital. Okay. So, if we look at the catabolism, since these are essential amino acids, there is no synthesis. So, we will only see how they are broken down in the system. It involves multiple steps. Okay. Each amino acids undergo their own way, got their own way of being broken down, but the first three metabolic reactions actually are common. Alright. The first three type of metabolic reaction are common, and in some cases the enzymes are also common. So, the first step is transamination, the second step is oxidative decarboxylation and the third step is a dehydrogenation. So, all the three amino acids undergoes this reaction to start with, then they will be diverted into their individual metabolic fate. So, when we are discussing the first step, we already know transamination takes place by a transaminase enzyme. Where the transaminase enzyme is acting on? It is acting on the branched chain amino acid. So, the enzyme is named as branched chain amino acid aminotransferase. Any aminotransferase or transaminase enzyme requires a cofactor vitamin B6 pyridoxine in the form of pyridoxal phosphate. So, these fundamentals should be absolutely clear by now. Now, this has actually three isoforms. So, this transaminase for branched chain amino acid has got three enzymes depending on their site of action, and one is actually very specific for leucine. Both this enzyme is present both in the cytosol and the mitochondria. Also, the activity is very high in the muscles, right? And who increases the activity of these enzymes in muscle? Naturally, starvation, when we need to break down more and more branched chain, right. When muscle is being degraded to produce energy, then this enzyme is reduced and the process of catabolism of branched chain amino acid will starts, right. So, this is the transaminase enzyme. Mind it, I have the slide has been designed in such a way so that each and every amino acid will be branched chain amino acid and their corresponding product will be shown in different color, right. So, the black corresponds to the products of valine, blue for leucine and green for isoleucine for your easy understanding. The first step, what happens? They are converted to their respective keto acid. Very fundamental rule, any transamination amino acid will be converted to a keto acid. The name of those keto acid: alpha keto isovalerate for valine, alpha keto isocaproate for leucine, alpha keto beta methyl valerate for isoleucine. Alright. If you are finding it difficult to remember the names, you can choose to omit, you can choose not to remember these specific names because the most important thing to remember in this branch chain amino acid metabolism are the enzymes. Okay. So, after the first step of transamination, the second common step was oxidative decarboxylation, right. It takes place by the help of an enzyme dehydrogenase. It is acting on branch chain keto acid because keto acids are produced in the earlier step. So, this is known as branched chain keto acid dehydrogenase. This enzyme is present in the inner mitochondrial membrane, and in mechanism it is very similar to pyruvate dehydrogenase complex, which was taught in carbohydrate metabolism, which was converting pyruvate to acetyl coenzyme A. Similar enzyme alpha ketoglutarate dehydrogenase complex, right. So, those enzymes were having actually 3 enzymes and 5 coenzymes same. This has also got dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase along with 5 coenzymes TPP, lipoamide, FAD, coenzyme A and NAD. So, multiple vitamins in their active form are playing a role in this step. So, what happens? Alpha keto acid dehydrogenase, this is branch chain alpha keto acid dehydrogenase, converts their keto acid to respective acyl CoA thioesters. Okay. This is a regulatory enzyme and its activity is increased by dephosphorylation. Mind it, since the mechanism is same, PDH was activated by insulin, dephosphorylation, alpha ketoglutarate dehydrogenase again by dephosphorylation. Similarly, this enzyme is also activated by dephosphorylation, very important, although it is a catabolic enzyme. And more active form occurs in the liver in well fed state and muscle during starvation. Muscle during starvation that we already know, right, but in liver it is in well fed state where the activation occurs. So, what happens actually? The keto acids are being converted to their respective thioesters. What are the names? Isobutyryl coenzyme A, isovaleryl coenzyme A and alpha methyl butyryl coenzyme A. Okay. This reaction, as I discussed, requires 5 coenzymes, but remember NAD and coenzyme A must. Okay. Also TPP is required, thiamine pyrophosphate, vitamin B1, right. This enzyme is very important because it is lacking in maple syrup urine disease. We will be discussing it very soon, but remember the name of this enzyme, branched chain alpha keto acid dehydrogenase. Okay. Next, dehydrogenation, another dehydrogenation. Okay. So, what happens? Oxidation of these thioesters are formed in the oxidative decarboxylation step leads to double bond formation or alpha beta unsaturation, right. It is analogous to the FAD link dehydrogenation in the beta oxidation that was taught in the fatty acid degradation. You know, whenever we are discussing about dehydrogenation, we should know a concept. Dehydrogenation does what? It introduces double bond, it takes up hydrogen molecule. So, this will become this, alright. If this is an NAD dependent dehydrogenase, what happens? The NAD forms NADH2, the hydrogen is removed from the same carbon. Whenever it is an NAD dependent dehydrogenase, the result will be the same, double bond will be formed, but the same carbon atom will lose 2 hydrogen. Whereas, if it is FAD dependent dehydrogenase, what will happen? The hydrogen will be lost from adjacent carbon atoms. Okay, this is the reaction mechanism that happens. Okay. In this case, these are moved from adjacent carbon atoms and this leads to an alpha beta unsaturated acyl CoA derivative. What are the names of those derivatives? They are methyl acrylyl CoA, beta methyl crotonyl CoA and tiglyl CoA. So, this is actually the third or common reaction that is taking place via the FAD dependent dehydrogenase. Mind it, in fact, individual enzyme can be named just like this. So, for example, if we are degrading isoleucine or leucine, it will be isovaleryl CoA dehydrogenase, that will lead to formation of beta methyl crotonyl CoA. So, as you know, enzyme can be named after a substrate on which it acts. So, dehydrogenase was acting on that compound. So, that dehydrogenase forms beta methyl crotonyl CoA. Why I am saying this? It is important, we will see in the disease section. Now what happens after these 3 common reactions, these individual amino acids go to their own path. Each individual amino acid suffers a different metabolic fate. In case of valine, it undergoes conversion to beta hydroxy isobutyrate, which again undergoes a dehydrogenation to form propionyl CoA, and we all know when propionyl CoA is found, how it can be converted to methylmalonyl CoA and then succinyl CoA and ultimately to glucose via the neoglucogenesis and carbohydrate metabolism pathway. Next we move to leucine. Leucine has got a different fate where it undergoes a carboxylation to form beta methyl glutaconyl CoA and ultimately it forms HMG CoA or hydroxymethyl glutaryl CoA. You already know the name HMG CoA, you have read it in cholesterol synthesis, ketone body synthesis, where you have been taught how HMG CoA lyase enzyme acts. We will see in the next slide. And in case of the third pathway, what happens? Tiglyl CoA is again converted to alpha methyl beta hydroxy butyryl CoA, then it is converted to alpha methyl acetoacetyl CoA by dehydrogenation. Okay. So, I hope this slide is clear. Again, let me tell you, you do not need to remember these individual steps. Okay. In the grand scheme of things, when you are revising for the entire curriculum, this is not so much important compared to the disease section, because disease section is a must know area in this branch chain amino acid metabolism. This is nice to know. If you can answer it, you will get more marks in Viva, or you may answer a very tough question. But if you miss the disease section that we will be discussing later, it will be problem, because that is a must know area. So, after this step what happens? The HMG CoA is acted upon by HMG CoA lyase. This we already know how via 3 steps. Carboxylase, racemase and the mutase, it is converted to succinyl CoA. You already know that. Here beta ketothiolase, it breaks down this alpha methyl acetoacetyl CoA to form both acetyl coenzyme A and propionyl coenzyme A. Okay. This is a recap of last class what we discussed, how propionyl CoA is the fate of propionyl CoA, how propionyl CoA is converted to succinyl CoA, which was also taught in beta oxidation of odd chain fatty acid, right. Anyway, so if we look at the fate, valine enters exclusively in the glucogenic pathway, leucine exclusively enters the ketogenic pathway, because acetoacetate and acetyl CoA both are precursor of ketone body, they form ketone bodies, right. And this enters glucogenic pathway by propionyl CoA as well as ketogenic pathway by acetyl coenzyme A. So, very interesting. So, considering the interrelationship of amino acid, this diagram was taught in last class while we discussed how methionine and threonine was being converted to succinyl CoA, and I told there will be new players that are joining. Well, those 2 players are valine and isoleucine, because both are converted to succinyl CoA. Okay. So, if we just magnify it a bit, valine via multiple steps, 7 steps to be precise, is converted to propionyl CoA, and isoleucine via 1 step less, it converts to acetyl coenzyme A and also propionyl coenzyme A by the beta ketothiolase enzyme, and we all know how propionyl CoA leads to production of succinyl coenzyme A. So, looking at the metabolic fate where our branch chain amino acid metabolism or branch chain amino acid are lying, you see isoleucine and leucine are forming acetyl coenzyme A, leucine is exclusively forming acetyl coenzyme A, whereas isoleucine is also forming succinyl coenzyme A along with valine. All right. So, we have discussed how branch chain amino acid enter into the TCA cycle, or they enter or they have a ketogenic fate. All right. So, this is the final common pathway of carbon skeleton where our today's amino acid have also been addressed in this piece of puzzle. Now, we move on to the various disorders of branch chain amino acid metabolism. The first disorder, which is an inborn error of metabolism, very important, is maple syrup urine disease, MSUD, right? Why it's called? It's first of all because the urine of the affected individual smells like maple syrup or burnt sugar. All right. You see, if you are on, maple syrup may not be available in our country, although it's available now because more westernized food are being available in shopping malls, etc. Right. But burnt sugar smells very similar, right? We don't have maple tree in India, right? Anyway, and the enzyme that is deficient, I told you, that is branch chain alpha keto acid dehydrogenase, which was converting the keto acid into their thioesters. This enzyme is deficient. So, what happens? I already told you, the blockade leads to hamper of conversion of these keto acids into their respective thioesters. So, what will happen? Both the branch chain amino acid and their corresponding keto acid will be elevated. The first step was amino acids all being transaminated to keto acid, and the next step, these keto acids were being converted to their esters. If the esterification does not happen, both the two previous steps, the intermediates, substrates will be high. So, that is the reason why plasma and urine concentration of branch chain amino acid and their keto acids are highly elevated. Okay? And since these keto acids are excreted in urine, especially the branch chain keto acid, they are also known as branch chain ketonuria. Now, this disease, that is classic maple syrup urine disease, is actually referred to the term where there is the maximum absence of the enzyme activity of this enzyme branch chain alpha keto acid dehydrogenase. Okay, the incidence is 1 per 1 lakh birth, and in fact, it has been found that less than 2% of normal enzyme activity is present. That actually leads to a problem where these branched chain amino acid cannot be metabolized in any way, and if cannot metabolize, their intermediates will be accumulated in the blood. All right. So, what are the symptoms of classic maple syrup urine disease, which is also abbreviated as MSUD? So, accumulation of branched chain. Now, the symptoms are very common in any inborn error of amino acid metabolism. Whenever something is, or amino acids are cannot be metabolized, they will be accumulated, they will saturate the transfer, they will help in a brain development, there will be mental retardation, intellectual disability, abnormal movement. All these things that we already read in previous amino acid metabolism disorders will fit into this disorder as well. Okay. So, what happens? Impairment in transport and function of other amino acid because of accumulation of excess amount of branched chain amino acid. When other amino acids are less available, protein biosynthesis will be reduced. Not only that, this branch chain amino acid actually competitively inhibit the very important amino acid glutamate dehydrogenase. Remember, GDH was an important enzyme which was helping in transamination followed by deamination, that is trans deamination, removal of nitrogen group. If it is hampered, there will be ammonia toxicity as well, because ammonia cannot be removed from the system. Anyway, after birth, since these are inborn, this is an inborn error of metabolism, the deficiency of this enzyme or the problem in the BCKD gene is already present to start with, and the baby will exhibit subtle symptom to start with. What are those subtle symptoms in the first 1 or 2 days? That is poor feeding, either breastfeeding or bottle feeding. The baby will appear to suckle very poor, there will be lethargy and irritability, the baby will cry, it will failure to thrive will happen. There will be abnormal movement. If this is continued and the disease is not detected, what will happen? It leads to focal neurological signs. What are those focal neurological signs? What are those focal neurological signs? These are abnormal movement, for example, athetosis, hypertonia, spasticity, muscles will be very spastic. Okay, there will be increased tone. Opisthotonus. Opisthotonus means the posture that is found in tetanus, where if this is the head of the baby, I am drawing a schematic diagram, and this is the table, the baby will curve like this. Okay, on the table the baby will appear, this will be the hand of the baby, and mind you, the eyes are looking up. Okay, this is the baby. So, baby will be curved like a bow. This is also known as opisthotonus. It is found in one of the key features of tetanus, but this is also found in classic maple syrup urine disease if there is excess accumulation of branched chain amino acid and their keto acid in the system, and ultimately it can lead to convulsion and coma. And if MSUD is left untreated, the central neurological function will hamper and ultimately it will lead to respiratory failure followed by death. Okay. So, this is a very serious disorder which needs to be detected very early and it needs to be promptly treated, right. So, how we can diagnose? Diagnosis is actually very easy if you know the biochemistry and metabolism of it. Now, diagnosis of every disorder is very easy. Urine contains branched chain keto acids and also those branched chain amino acid like valine, leucine and isoleucine, alright. Rotheras test is positive. When does Rotheras test become positive? In case of ketonuria, mind it, right. You know, in classic Rotheras test, when we boil and cool the urine, the acetone and acetoacetate may get evaporated and that might not give a positive Rotheras test, but these branched chain keto acids are not heat labile. So, even if we boil and cool the urine, still Rotheras test will be positive. Okay. There is one applied importance that might help you in practical exam. So, Rotheras test, we got one answer from phenylketonuria, also maple syrup urine disease. These are the two disorders of protein or amino acid metabolism in which the Rotheras test is positive for ketone bodies, alright. Now, the diagnosis depends on enzyme analysis. I told you the level of the enzyme in classic MSUD, it is less than 2%, right. And very important, the diagnosis should be done prior to or 1 week after birth, right. Means if we let the baby undiagnosed, these symptoms are continuing and the baby, if it is detected prenatally, it is very good. If it is not detected prenatally and if we cannot detect even if 1 week has passed, by then the brain development is so much hampered that will lead to permanent mental retardation and it can also even lead to death. What is the treatment? Well, the culprit, since those are essential amino acids and their metabolism is hampered and they are not synthesized in the body, we can restrict their availability by designing a diet in which there are very low amount of branched chain amino acids. Mind it, every amino acid is essential for protein synthesis, right. But when they are not needed, these branched chain amino acids or every amino acid is broken down in their own catabolic pathway. So, very low amount must be given because it is essential for brain development, but not in excess, alright. So, a very important balance needs to be struck. There are other variants of maple syrup urine disease that are not severe. Intermediate maple syrup urine disease is one such in which the same enzyme is deficient. Okay. All maple syrup urine diseases are due to deficiency or inactivity of this enzyme or less activity of this enzyme, branched chain alpha keto acid dehydrogenase. In this case, 3 to 8% of normal enzyme activity is present. In classic, it was less than 2%. All right. So, what happens? Since it has got a greater level than residual enzyme activity in classic MSUD, the symptoms are generally late to appear, alright. In that case, classic, it was appearing within first 24 to 48 hours and if you do not treat by 7 days, the baby might die. Here 5 months to 7 years, because till then the whatever residual level of branched chain keto acid dehydrogenase activity is there, it will help to normalize the brain function. Mind it, whenever there is more enzyme activity, the diagnosis will be much later because there will be less problem. Whenever there are severe enzyme deficiency, the diagnosis will be earlier, right. So, it depends on the amount of enzyme that is deficient, right. The symptoms are actually same as that of classic MSUD, but generally milder and they appear late in case of intermediate MSUD. Further lighter variant is known as intermittent maple syrup urine disease. It is also known as intermittent branched chain ketonuria. It is a milder variety because 8 to 15% of enzyme activity are present. Okay. So, What happens? Contrary to classic and intermediate MSUD, alright, these individuals have got normal growth and normal intellectual development. Why? Because the enzyme activity with 15% enzyme activity, generally there is no problem, alright. So, it will not be diagnosed unless there is a crisis situation, when there will be muscle degradation and when there is increased catabolism of branch chain amino acids, only then this will lead to a problem, right. This is in case of excess stress, in case of starvation, or in case of infection, when there is an increased catabolic state, when excess muscles are being degraded, then only when excess branch chain amino acids will be degraded, then only the feel of the absence is, the absence is felt, alright. So, metabolic crisis leading to seizures, brain dead is still a possibility, it may happen, but if the disease is diagnosed and careful diet is planned, so that after diagnosis, suppose in case of a crisis these symptoms happen, alright, but after the crisis is over, if a dietitian gives specific foods which are less in these branched chain amino acid or special formula feed, then this disease will be taken care of. There is a very minor variant of maple syrup urine disease, that is also known as thiamine dependent, okay, because that is similar to intermediate, right. It is because even if the enzyme is not active, right, somehow it the function can be improved by excess dose of vitamin B1 or thiamine. Why? Because the enzyme branch chain keto acid dehydrogenase needs thiamine as a cofactor, alright. So, large dose of thiamine will increase the activity. Generally, if the thiamine responsive MSUD is present, newborns do not present as a, this does not present as a disorder. So, newborns rarely present with symptoms, but whenever the baby is growing up, for example 5 to 8 months or even 5 years, where intermediate MSUD was being diagnosed, first we can actually check by dosage, excess dose of thiamine, and that actually helps in treating the disorder. After MSUD, there is isovaleric acidemia, which is nothing but specific inborn error of leucine metabolism. The first 3 reactions were common, and the first was transamination, the second was dehydrogenation, and the third one was FAD dependent dehydrogenation, right. The problem lies in the reaction 3 of leucine metabolism. So, what was the enzyme? In the reaction 2, isovaleryl CoA was produced, right, and this isovaleryl CoA cannot be further metabolized. So, isovaleryl CoA dehydrogenase is deficient, it was converting it to 3 or beta methyl crotonyl CoA. This conversion cannot happen. So, what happens? Isovaleryl CoA in the form of isovaleric acid is accumulated in the body, and its level is very high in sweat and urine. So, whenever, that is a common theme, whenever an intermediate is accumulated, its level will rise in sweat and urine. But a characteristic how we can diagnose it is this acid is secreted in the body fluid and it imparts a cheesy odor to breath and body fluid. Okay. And the symptoms include acidosis, lethargy and mental retardation, that is the same to all amino acid intermediates. If they get accumulated, that is leucine further cannot be accumulated, it will the concentration will rise in brain and it will lead to hampering of other amino acid metabolism, leading to mental retardation by those keto acids that were being accumulated in MSUD from leucine is also getting accumulated in isovaleric acidemia, right. So, the symptoms are more or less the same. And lastly, hypervalinemia, this is a disorder, again an inborn error of metabolism. What happens? The transamination of valine is selectively impaired, the first step impaired. Again very common, what will happen? High plasma and urine valine level, but since it is selective valine transamination defect, leucine and isoleucine level will remain normal. But again the clinical feature it is similar, growth failure, hyperkinesia, abnormal movement, vomiting. Nystagmus means rapid eye movement, to and fro eye movement, it can be up down, it can be horizontal, there is a nervous CNS sign, and inability to suckle, that is irritability to breastfeed. So, this are the disorders of branch chain amino acid metabolism. To conclude, this lecture session has covered the chemistry of branch chain amino acids, their metabolic fate. We learned what are the steps of catabolism of branch chain amino acid. We learned about the disorder, maple syrup urine disease, most important. We learned about the variation of maple syrup urine disease, the symptoms, the diagnosis, the treatment. Also we learned about isovaleric acidemia and also hypervalinemia, which are selective defects in the metabolism of leucine and isoleucine and valine. So, these are my references for today's class. I thank you all for your patient hearing. Bye.